So it's a privilege indeed to present and to recall my history with Roy and history with Pedro and Leslie and Talia and many others, Jorge. I was a graduate student at the University of Waterloo, and I read Roy's 1967 book, Mechanisms of the Brain, as a young man. And was very impressed, especially with the review of the Russian science and the history of that. And I designed a dissertation that had over 200 rats with 4 to 5 electrodes implanted, doing a conditioning paradigm where I would flash. Essentially, they would go down they would just hit a bar to eat pellets. And then one day for half of them, I would present a 3.3 cycle per second light, at the end of which they would receive a foot shock. And the other half, randomly selected, where I would present an 8.2 cycle per second light, followed by a foot shock. There was 4 groups of 40 seconds. Start my video.
And Dr. Thatcher, thank you. Yes. Now we
Okay,
can see you. We can see you now. Thank you.
Yeah, a little organizational issues here. And so those 2 groups of conditioned with different frequencies, then the next day I would present a light halfway between the 2 at 5.8 hertz. And the animals that were conditioned at 3.3 hertz, you would see 3.3 hertz rhythms coming out of the brain. Those at 8.2 hertz, you would see 8.2, and the subharmonic 4.1. And then I had 40 seconds, 60 seconds, 80 and 120 seconds CS UCFS interval. And after 60 seconds, rats generally have a difficult time retaining the memory, but I was able to see the memory trace. I shared that with Roy over the internet, or at least at those days by fax or something. And he then had Frank Morell. I helped, was part of a program there as a senior at the last year of graduate studies for a research seminar series. And Frank Morell was working with Roy at New York Medical College at the time, and he came to Waterloo, and it was great to meet him. Dominic Purpura from Albert Einstein College of Medicine also came. And I ended up going to Albert Einstein College of Medicine as a postdoctoral fellow in 1971. But I interacted with Roy at that time, and I had a choice to go to New York Medical College as an assistant professor, or stay at Albert Einstein doing intracellular impalements and 1-day-old to 3-week-old kittens. And I chose EEG and went to New York Medical College, and I'm very happy I did. And Roy was fantastic. One of the amazing things at the time at Albert Einstein College of Medicine was that Eric Kandel was interacting, and in which it was evaluating the dynamics all the way from RNA to protein to synapses, and for which he was awarded a Nobel Prize in the year 2000. He evaluated primarily aplasia and looked at serotonin and dopamine, and could see how it changed the size and number of synapses, which was profound. Then I joined Roy, as I mentioned, in 1973. Roy and Paul Easton went to Switzerland, I believe, to meet with Matusik and Peterson, who had the first normative database. And that was an analog database, and we got copies of that. And then we start replicating it. And then, of course, we had digital computers that were on the scene. And so we started doing spectral analyses to create our own normative database at New York Medical College. Roy and I began working on this book, Functional Neuroscience, Volume 1. And by the way, Volume 3 was with Talia and Pedro. So thank you both. So we edited volumes 2 and 3 and wrote this book together. On chapter 12, if you ever can get the book, it's available on our website. It's free. You can download it. Roy had a whole section on mental experience and the physical basis of consciousness. and he was thrilled, just engaged by the concept of consciousness. And this is the earlier stages of his evolutionary education on consciousness that just became more and more sophisticated over time. But it's good to see it. I then went to University of Maryland and collaborated with Roy. Roy was kind enough to ship. At that time, we had a large National Science Foundation grant where we had portable trailers, essentially, that had EEG amplifiers in it that we'd move around in different parts of the country, one of which I was able to arrange to come to the University of Maryland. And some of the data that I collected, part of the multi-million-dollar grant there, was included in Roy's normative database, a little bit of it. But Roy and Leslie visited me in Baltimore, and we worked together. But essentially, this is the procedure that I developed, my team developed, to make sure that we had Gaussian distributions that met all the fundamentals of statistics, so that you can get cross validations that made sense for a variety of different areas, including three-dimensional imaging of the sources. But at that time, it was just at the surface. People did not understand very much in 1979, 1980, that much about the sources. This is the normative database that we developed from birth to 83 years of age. It has over 730 or 750 subjects, very carefully screened with neuropsych tests, clinical evaluations, etc., and over 1,500 subjects. Only 750 or so met those criteria. Then I went to the Bay Pines VA and University of South Florida, and continued working on EEG. But this time, and I discussed this with Roy at the time, but what we did is we used the MRI and calculated T2 relaxation time, and as well as EEG and different slices of the MRI. And we could look at gray matter, white matter, and we could correlate that with the power, as well as the coherence, the connectivity between different parts of the brain. And we had traumatic brain injured patients, and we had normal subjects. We had neuropsych tests, etc., and we got very high correlations relating the connectivity of the brain. And we also found that there are two compartments. The short distance compartment really it acts like a diffusion process. It's the small unmyelinated axons that connect local regions, and it's like if you get a felt pen and you put it on a paper towel, you'll see the ink just flows out. That's the short compartment. It has a particular exponential. We have the coherence equation there is e to the minus kx. That is the diffusion component. And then there's a long distance component where information goes, let's say, from the parietal lobes to the frontal lobes, and then from the frontal lobes back to the parietal lobes. So it's all in loops in the white matter, and that's the long distance compartment. And we could correlate that with the MRI on a large number of subjects. So we integrated the structural integrity of the brain with the power spectrum, as well as the functional connectivity. And they're very much related. We got highly significant effects, and we also had neuropsych tests. You could see that the greater deviation from normal, the lower on the neuropsych tests. At the University of South Florida, we did MRIs on these patients, many of the patients, as well as Alzheimer's patients. And healthy patients, you can see a very clear bimodal distribution between the gray matter and the white matter. But as Alzheimer's progresses in its severity, you just get a unimodal, just a single bell-shaped distribution. There's no good distinction between gray matter and white matter. All of that is very important because we could quantify it and we could relate it to the EEG power spectrum. In 1990, I went to the National Institutes of Health around 1990, 91. I was a project manager for the first 128 channel EEG and part of the human brain mapping program. And it was totally illuminating. This is like Disneyland for a scientist. It's just unbelievable what goes on at NIH. I walk down the hall and there's 6 PET scanners, and down the hall a bit more, there's a whole bunch of MRI machines, and there we had 128 channel EEG plus magnetic encephalography and SPECT scans and later diffusion tensor imaging and functional MRI. So this was the flowering moment in the 1990s for neuroscience. It was the human brain mapping program set up by President George Bush I. And he put $2 billion into it the first decade, and then another $2 billion the second decade, another $2 billion the third decade. But that's the human brain mapping program. And I invited Roy to help out as a co-author in this book on technical foundations, functional neural imaging. You may not be able to see it. And Roy made a great contribution, and Leslie as well. Statistical probability of brain mapping and function and structure in the book. That's chapter 13 for those who want to read it. Then in the same program there, as Roy was present, and we had discussions about this. This is the paper that I wrote, and we all worked together as Camilo Toro and Mark Flager and then Mark Hallett, who was the director of the National Institutes of Neurological Disorders and Stroke, is one of the main program managers at the National Institutes of Health. But I cite in this paper the findings the things that Roy and I talked about a lot, and Roy was very interested in, which was zero phase locking of coherence. And there's a number of studies that had shown this. Gray et al., Eckhorn, 1988-89, Murphy and Fetz, 92. And then Roy and I, in our book, we could see zero phase lag. And Roy was very interested in that in his 1963 paper too. And so I wanted to explore what it meant for there to be zero phase lag. Now, essentially, it was a mystery, but we could see that really what's going on is the thalamus is like at the center of the brain. And so it could synchronize parietal and frontal lobes at zero phase lag. Largely, that's how it happens. It's not through the white matter. It literally has coordinating hubs in the brain. And at NIH, we had imaging. We could combine the PET scan with the functional MRI and EEG and MEG, and we could see where it's going on between the thalamus and different parts of the brain. One of the things we did this time was to calculate using complex demodulation and later on the Hilbert transform instantaneous coherence and instantaneous phase. So you don't have to average, you can do it in very brief periods of time. And we put leads on the thumb. That's where the bottom row is, the EMG. And you could see when people would move their thumb, what the task was is a voluntary thumb movement. So when people just felt like moving their thumb, they would move their thumb. So there was no stimulus telling them to move their thumb. However, subconsciously, there's this buildup. You can see the BP potential a second before, 1.5 seconds before. Even though people were not aware of it, their brains were subconsciously getting ready. And then there was as soon as they decided to move, there was a huge flow of information from the supplemental motor cortex and other regions around there that we could see in the PET scan and the functional MRI, but mostly PET scan. And then after the movement, there's a second potential. You can see this PPMP. That's evaluating the success of the movement. This is similar to what was presented earlier. Their brain is continually evaluating. It plans things, it does things, and then it determines the success of whatever was done, and then corrections are made. So the next time you do it, you'll do it better. This is happening all the time. If you go to your cupboard to get some cereal, open up the cupboard expecting cereal to be there, but the cereal's not there. What happens, your nucleus accumbens is pushing out all kinds of dopamine in anticipation of this future reward. but it's not there. So your insula inhibits all of that. So you don't keep opening the cupboard all the time. And then you go over to the other cupboard, open it up. There's where the cereal is, and now your dopamine is happy in those parts of the brain, the nucleus accumbens, and you got a reward and you eat your cereal. So at National Institutes of Health, we did that same task, but we had 32 or 36 electrodes, and so we could derive dipoles in the brain, in this case, 3 dipoles, that would account for about 95% of the variance. And the supplemental motor area and the motor cortex were the main ones that came out in our analyses. And you can see the way the brain gets ready to move, and there's a buildup of, dipole 1, and then post movement, dipole 3 comes in, or one of the, I can't tell exactly which one. That's evaluating the success of the movement. We saw that in all of our subjects, but now we have the power to see this in great detail, including looking at instantaneous coherence. So we're using complex demodulation or the Hilbert transform. We could look at the coordination in milliseconds of time between these different dipoles, pre-movement, then immediately post-movement, and evaluation of the success of the movement. And so now we have the power with instantaneous coherence and instantaneous, so very short durations of phase differences, much greater precision in the network dynamics and hubs in the brain. Inspired largely by Roy wanting to be able to look at this in greater depth, and this is just summarizing the supplemental motor area and the motor cortex, the way they shift and change in preparation of the movement and then post-movement, evaluate success in the movement, etc. We saw this in all of our subjects. But you can do it with instantaneous coherence and instantaneous phase differences. And then we use what's also called the phase slope index, where you can look at the direction and magnitude of information flow. I don't know if you can see this, but this is Roy's paper. Let's see if I have a hard time to move this down. Back up. This is a paper that Roy, Paul Easton, and Bob Eisenhart wrote, Consciousness and Cognition, but may be mediated by multiple independent complement ensembles, in the journal called Cognition, Consciousness and Cognition. And the editor of the journal contacted me and asked me if I would write a review or evaluate this very interesting paper looking at working memory that Roy did, and his collaborators, with emphasis on distributed cell assemblies and coherent synchronization in preparation for a movement and then evaluation of that movement. I thought it was a very good paper, so I reviewed it. This is part of my review. I titled, though, Neural Coherence and the Content of Consciousness, because Roy and I were in a lot of discussions at this time about the general operation of consciousness. That's when you sleep and you wake up, or you have arousal or a coma. These are general operations. And then there's the specific operation of consciousness, which is where you are aware of where you are and the content of your perceptions and of your feelings. And, so the content of consciousness versus the general operation of consciousness. These are topics that Roy and I discussed a lot. And so I put this into this chapter, and I evaluated the zero phase locking of coherent activity that Roy and colleagues and others that I mentioned before, Singer and Gray and Eckhorn, Murphy and Fetz, and also John Morgadis and also Alexis Ramos with Roy and Eric Schwartz, saw this zero phase locking all the time, and which was very inspiring, very interesting. And so I wanted to elaborate on this a little bit more. And so I evaluated this general operation of the conscious of the binding process and the content of consciousness. And I'm not going to go into a lot of detail, but I do at least show you some of the thoughts that arose by discussions with Roy and thinking about this over periods of time to try to create a model that is can account for the content of consciousness and underlie what's going on with finger movements and the intent to move, and why phase shifting is involved in it and phase locking. Now, that's the other thing we found at the National Institutes of Health is there's always a phase shift. It's usually 40 to 80 milliseconds. The longer the duration of the phase shift, the higher the IQ. Children with severe autism had phase shifts only 20 milliseconds or so, and then they had phase locks for over a second. So what's going on is the brain is shout out saying who's available at a given moment of time, because neurons they may be refractory, they may be engaged in other tasks and not available, or they just may be exhausted out of the several billion in a given Brahman area. And so there's a phase shift that's a recruitment. When I worked at as a 11th and 12th grade in Long Beach, California, I would go to the shipyards to make some money in the summers or whenever I could, going on boats and unloading things. But if you get there early enough in the morning, you're sitting in the back, the guy comes out and says, "Who's available?" I raise my hand, and then I'd get recruited. I go to the boat, and I get a job done. If you come out there too late, you're not available. So it's availability, phase shift, who's available. Those neurons get recruited, then they become phase locked to mediate a task for 200-300 milliseconds in healthy brains. That is now well understood. And so I went ahead and developed a nonlinear dynamical model. I'm not going to go into a lot of detail about that, except that there's always interactions between oscillators, and that there's weighting functions that determine how long these oscillators will be phase locked and what the duration may be of a phase shift. And so you're able to put this in precise numerical mathematical forms using the equations that Baldi and Mayer developed, and Cohen and Copeland and Herman Trout did. And so those equations fit for neural network dynamics. Look at the recruitment both for short and long distance coupling. And we have these two dynamic models that are going on continuously in our brain. And we have enough precision now with EEG using instantaneous measures of power and coherence, et cetera, to use differential equations to fit to these data. And so I went ahead and elaborated on that. I'm not going to, this is available on my website if you want to read the whole paper, but eventually came up with a slight modification, as you can see in equation 7, that helps describe all the data that we could see in our studies using the PET scans, functional MRI, and the inverse solutions. And at that time at NIH, we evaluated about 10 different inverse solutions. The one that Pedro presented is one we looked at. Roberto Pascual Marquis is one, but there's a whole bunch of them. We looked at the accuracy, and one thing we found is that there was fairly good accuracy of source localization at the crest or the crown of the brain, but when it comes to the temporal lobes, there wasn't. And that's because the brain is not spherical, it's shaped more like a loaf of bread. It's elongated and flat on the bottom. So if you use a spherical head model, you'll always have error. And S. Loretta was a method that Roberto Pascual Marquis used to try to minimize that error using a spherical head model. But the National Institutes of Health, we use a realistic head model. That is we would actually trace out the boundary of the cortex on the MRI in every subject, and no longer used a sphere. We used a realistic head model, the actual geometry of the brain, and we dramatically increased the accuracy. And today, the databases that are used to evaluate patients from the Montreal Neurological Institute database and the National Institutes of Health this is the MRI, the MRI from Montreal Neurological Institute, as well as the MRIs from the National Institutes of Health, are all using the realistic head model, the boundary element method it's called, to look at the boundary and then solve the inverse solution with greater accuracy. And so by doing that and looking at it in details, I was able to come up with this general model where there are preferred pathways and non-preferred pathways, and these are somewhat binary for excited regions and non-excited regions, but also preferred pathways. So you can have excited regions, but they may not be preferred. They may not be connected. They may not be adaptive. If you want to move, it's got to be the frontal and the parietal to do that, etc. You need those neurons to be excited and communicated, and you can kind of come up with this model I use to account for finger movement and looking at phase shifting and phase locking. That's a type of a labeling process as an extension what Roy had been talking about for a long time. He was always interested, and then this is a conclusion I make to summarize Roy's thinking about, but extend it more to the content of the present, and also to note that in 2002, Roy he was the book that Roy and I wrote together. He had an extensive chapter, or a very expansive. Now, it wasn't extremely expansive, but it was penetrating intellectually about the content of consciousness, the operation of consciousness. He had quite an interesting model he really later abandoned, but he stuck with most of it that was presented earlier today and your various presentations that were so excellent. So anyway, that's my talk, and I want to thank you for inviting me.